I would like to pick up your tests either now or before the end of the hour, so whatever you'd like to do, we can pass them in. Or all right. Thank you. We were speaking about the chemotaxonomic aspects, and we had to do that very hastily, so I think I might clarify a couple of points, but I want to spend most of the period on the grapes. The points on page 2 of your notes, some generalizations from genetic studies point out a couple of interesting things that will relate to our discussion of grapes. For instance, item 2 says a diglucoside is dominant to the monoglucoside, at least as far as anthocyanins go. The typical glucosidation or glycosidation pattern in anthocyanins is in the 3 position first and then the 5 position next most often. And the point is that the diglucoside condition seems genetically dominant in most plants, and that is the case in grapes, and that'll become important when we talk about grapes just shortly. I think the rest of these comments, the acylation is generally dominant to non-acylated. That also has some relationship to grape varieties that we will mention. A number of these points are fairly obscure unless you're going to be interested in this area. I think a couple of them became obvious in the course of your take-home test. For instance, on item 10, page 3, it mentions that hydroxylation and 9 and 10, hydroxylation and addition in the ring is more complex in terms of genetic evolution. And the hydroxylation in the anthocyanin series is related to that in the flavonol series. In other words, if you have a delphinidin anthocyanin, you would expect a myricetin flavonol in the same material. Again, that'll have some bearing when we talk about grapes, if I don't forget to mention it, but so it won't be obscure, I might bring it up now. For instance, in white wines, myricetin has not been reported. But in red wines it has, and certainly red wines contain delphinidin derivatives, so this may be indicating a genetic block further down the flavonoid production, so that the trihydroxy form, which is not needed for any other component in the white series, is lost along with the ability to make pigment entirely. Under B, as we suggested you read, it says it's 6,8 methyl flavonoids. Now that's not methoxy, but methyl directly on the A ring is rather characteristic of pines along with the still beans. And you had one example on your test. The substitution of hydroxyl groups in the A ring in 6 and 8 position, and in the example given on your test, further methoxylation then is rather characteristic of citrus, and so on and so forth. One I want to mention on that page that otherwise might not be clear, we won't talk any more about beta-cyanins, but you notice at the bottom of page 3 there, the structure of a beta-cyanin. Beta-cyanin itself is given, and this is a nitrogenous substance, as you see, with a positive charged nitrogen, which behaves very much like the positive charged oxygen of the oxonium series. And it's quite interesting, although this chemically appears to be quite different from the anthocyanins, in the plants where it occurs, and the beet is the common one, your ordinary red beet that you eat has this kind of compound, and that's where they get their name. Beta is the generic name of beets, so the beta-cyanins are common in beetroot and in other related plants of the centrospermi. Well, the point that's interesting is in the plants that produce these compounds, as different as they appear, they do not produce anthocyanins, so that these compounds replace the anthocyanins and have very similar colors, so that the need for a red color seems to be met. Just exactly what that is a little hard to know in this case, but whatever it is, the loss of the anthocyanin and the production of the beta-cyanin is a rather interesting case. Isoflavones are common in legumes along with the rotinoids, which are derivatives of isoflavones, and also legumes are noted for many other phenols, particularly coumarins and furanocoumarins. And again, some of these things came up on your take-home test, so I hope you're able to fathom that without too much trouble. The Ellagic Acid comment was made very briefly so that you'd have it for the test, but I would like to elaborate just a little bit. It's been shown that Ellagic Acid is generally absent, if not, we believe, completely absent, from non-vascular plants, which would be fungi, lichens, algae, that sort of thing, and also the ferns, which are vascular to a degree, the gymnosperms, and the monocotyledons. Well, now the monocotyledons are fairly advanced, evolutionarily fairly advanced plants. They're spermatophytes and somewhat up the scale in terms of higher plant evolution within the spermatophytes. And they think that this is, as I think I mentioned last time, a hapactic mutation, which means that it's one that it occurred only one time. And it apparently occurred, that is, the monocotyledons then apparently diverged from the rest of the dicotyledonous types before the rosales. So if we're making a tree of whatever, the rosales have ellagic acid and the monocotyledons don't. So apparently the monocots diverged from the general scheme of evolution before these and near the ranales because they lack. Now, I'm no great shakes as a botanist, so I'm not, these are the rose-related plants, but I'm not exactly sure. The ranuncula and a few others go here, but I don't know too much about the botanical point. But then the point is that the monocotyledons must have diverged somewhere near the ranales because they lack ellagic and seem to be on an evolutionary scale, and the rosales have it. So it would have to be in that general phylogenetic location that the mutation took place that occurred once apparently, and the ellagic acid appears in everything after that, at least of the woody types, and not before that. Now, why is that important? Well, we haven't made an issue of it yet in any great extent, and we can't carry it too far, but we have, I think, mentioned that the Gallic Acid analog in the Cinnamic series is absent in nature. And if we look at the common types of phenols, just giving, say, the anthocyanin series, which would be the monohydroxy down through from pelargonidin to malvidin, or in the Cinnamic group from paracumaric to synapic, Gallic is listed in parentheses because, of course, it's not a cinnamate, so that the trihydroxy B ring type is known, and yet the cinnamic that would appear to be necessary to produce it is not known. And the hydroxylations from 1 to 2 seem to be easy, but from 2 to 3 seems to take a separate kind of enzyme system that we don't know much about yet. So where did the Gallic acid cinnamic analog go? Maybe it's just so labeled that it falls apart and we can't find it, but people have looked, and that doesn't seem to be the case. It would be fairly unstable, but so are the caffeics, and they actually stand isolation quite well. Well, Bates-Smith has proposed that ellagic acid is the carrier of Gallic acid. So the reason you need a cinnamate group is that you need to contribute that B-ring type structure to further metabolic products. And Bates-Smith proposed ellagic on the basis of the chemotaxonomy of the thing. In other words, ellagic occurs in the plants where you would need to have a Gallic acid derivative and not in the other plants where you would not. Well, we've gotten a hint from grapes that I'll mention shortly that may not be the right reasoning, but it is true that if you want to make ellagic acid in the laboratory, it's quite easy to do. If you take Gallic acid and try to make ellagic, It's very difficult. That doesn't happen. But if you take methyl gallate, you can get ellagic very easily. And all you have to do is blow air through the solution in a weakly alkaline solution. So an ammonia, make the solution alkaline with ammonia, and methyl gallate will go to ellagic acid just by air oxidation. So in other words, it works very well in the test tube. Why wouldn't it work something like that physiologically? So the point would be then that esters of gallic acid could produce ellagic. Therefore, the occurrence in the plants that needed a synamic analog of gallic wouldn't necessarily be related to the presence of gallic, but might be related to the presence of esters of gallate, which would give ellagic easily as well. So in other words, the ellagic would be an artifact, but it would show up in the proper places as if it were a precursor. But maybe the real precursor would be an ester of the gallic acid. Well, what esters of gallic acid occur fairly widely? Epicatechin gallate and epigallocatechin gallate occur rather widely, and particularly in tea and grapes, they're major components under some circumstances. And we'll come back to this briefly. It's still a hypothesis. We can't prove that this is the source. Well, we can with the radioactive studies, but it hasn't been done yet that this is the case. But in any case, the ellagic does seem to serve as an indicator of this kind of situation. Another argument for it, not the precursor of further biochemistry not being ellagic, would of course be the carbon-to-carbon linkage that you'd have to break. So forming the carbon-carbon link may be fairly easy, but breaking it again to split the ellagic down into its component gallic acid units seems to me very unlikely. Plus, as we've already mentioned, the extreme insolubility of gallic acid once formed. That's why it's a nuisance of an instability type in Loganberry wine, is because it does precipitate and is so insoluble. Another point that I wanted to elaborate, or well, mention, it hadn't been mentioned before, is that there is an interesting, I don't know exactly what it means, but there is an interesting occurrence of the 4 flavanols, or flavan-4-ols of the type and the one with the B-ring-like cyanidin is this one. And it's called luteophorol. They use an F, yes. And luteophorol has been found in aquatic plants, such as water lilies and plants that grow from a lake bed up to the surface and do have roots underwater, which, except for rice and a few other plants that are specially adapted, is a rather uncommon way for plants to grow. Plants usually need aerobic metabolism to take up minerals and so forth. In other words, the roots must grow aerobically. So those that can grow with a waterlogged root system seem to produce this kind of compound as a group more than do other kinds of plants. Now, why should that be so? We haven't any idea, but it is true that these tend to be found in that class of plant. Thank you. It also is true that it is found in a few others. For instance, this compound has been found in sorghum, so it is found in a few plants other than the aquatic. But the point that I wanted to make is that if you then dehydrate this compound and produce a double bond in that position, then you have the 3-deoxy or desoxyanthocyanin type molecule, and this one in fact is luteolinidin, and it does occur in nature. And remember that we said that that was a primitive character. The anthocyanins without a 3-OH group, which of course can't have 3 glycosides then and so forth, are found in mosses and some early plants. Well, the idea that aquatic plants developed early and then migrated to the land is rather an interesting speculation in this connection. So these are some examples of the kind of thing that may be considered pretty hypothetical, but is very interesting. And as we learn more and more about this sort of thing, we are building a better evolutionary understanding of plants and a better classification scheme. In most cases, the classification schemes still depend largely on observable properties, leaf positioning and all this sort of thing that have been used for years in plant classification. But on the other hand, combining some chemical data of this type is working out some problems where people were unable by visual classification to characterize the plants properly, and then made use of this kind of information. Of course, phenols aren't the only ones, and some of the most interesting are probably going to be the amino acid sequence. So you've probably seen, there have been some very interesting articles in places like Scientific American on protein evolution in mammals, for instance, hemoglobin protein, showing that man is very closely related to the chimpanzee, which we knew all along, and not very closely related to the horse, and so forth, building up a scheme of evolution as to how closely related different creatures are. So in the proteins is where we'll eventually get the problem completely solved, if we ever do, but the phenols are certainly contributing. And it's also giving us some ideas of potential metabolic roles. Now, we have no idea where the 4-hydroxy compound fits into metabolism, but it does seem significant that this kind of compound is produced in a plant, a group of plants that are exclusively confined to a somewhat today rare type of growing situation, rare for higher plants. Any comments or questions about the chemotaxonomy? And since some of you have arrived since, if you now want to raise any comment or question about the test before we'll discuss it after I look it over and I'll say grade it. I will grade it. On the other hand, most all of you are going to do very well, I'm sure, because it's the nature of take-home tests. Do you have any comments or questions about either chemotaxonomy or the test itself that you'd like to raise at this time? I'd like to raise one. Since it's fresh in your mind, did you feel that it was unduly lengthy and time-consuming, or did you feel it was reasonably fair? Reasonably fair, I guess. I see most head nods in that direction. Okay. It's the kind of thing, of course, that there's no limit to how much time you can put on it, which puts the burden on you. But in a way, I think it's kind of good because it's rather typical of a quasi-research type thing, whereas it depends on how bad you want an answer. You can find it if you look long enough, but if you have the right way to find it, then it's not too long to do. Well, then let's go ahead with talking about the phenols of grapes. We have here 4 references that are recommended. Of course, naturally, I tend to feel that the reference A is the better of the series. And reference D is excellent, is the best thing yet on the astringency of fruits, and has one of the best comparative discussions of phenols in edible fruits of several kinds. C, the book by Hume, is quite good in some regards. In the general sense, I think it's a 2-volume set. Maybe more volumes are coming out, but right now it's a 2-volume set. But in the second volume, there is a chapter by some French workers on the phenols of grapes and wine, which I don't recommend. I find it's rather poorly done, in my opinion. So I'd just assume you didn't bother to read that one, or if so, read it for comparison, not for authoritativeness. First, we want to divide the discussion into 2 major subgroups. First of all, the qualitative occurrence of phenols in grapes and wine, and then the amounts, and then following that, something about the significance and what comes from the grape into the wine. The qualitative composition, as has already been mentioned in your notes, of a given plant is pretty well set by the genetics, and we've implied that in what little discussion we've given to the chemotaxonomy story. In other words, a plant of a given genetic makeup, the phenolic qualitative composition is fairly rigidly set by the genetics. so that it is not commonly found. If you do a, well, let's just for example, if you do a paper chromatogram of the phenols of a plant, this happens to be grape seeds, you wouldn't expect to find that if the weather was different, that spot would disappear. What you would expect to find related, say, to weather is that the whole group would gain or lose in amounts, but not whether or not a given substance is present. There are minor exceptions that are fairly, in the cases where known, where fairly explainable. For instance, it may take a, well, if a certain compound is light sensitive and you deprive the plant of light, then it's not going to be able to produce that compound. But in any sort of normal growing where we're talking about field variations, then you wouldn't expect the appearance or disappearance of a given substance based on environment. In a qualitative sense, if component A is there, then B ought to be there too if it's the normal circumstance. And not only that, but if A is generally 30% of the total and B 20% of the total, then that ratio will stay about the same, even though the actual amount may vary, at least within limits. Now that's a little less rigidly set by the genetics, but the overall amount of the phenolic group is quite sensitive to the environment. So that generally the genetics rigidly sets the qualitative composition, less rigidly sets the relative quantity of each substance, and then finally has very little to do with the total amount. That's more sensitive to the environmental condition. Now on that slide and in your notes, we have listed by pretty much groups the phenols present in wine. And you might immediately see one of the first things to point out is, say, for instance, in the anthocyanin series, we just showed a slide where we said that this was the normal series except for pelargonidin. Now, pelargonidin would be considered a normal anthocyanidin. It's not found in grapes yet, never been found. And by the same token, the leucoanthocyanin equivalent to pelargonidin has never been found in grapes. But other than that, the series is normal. So the emphasis would be toward more hydroxylation and methoxylation, and in that sense, the grape would be a fairly highly evolved plant. And I think we've already mentioned that the grape is highly evolved being a vine, because vines, the evolutionary pattern seems to be from lacking lignin and upright growth ability toward lignification and tree-like or shrub-like character, and then back away from this through vines toward the herbaceous monocotyledons and the herbaceous low-growing plants, so that in the evolutionary sense, the grapevine then is a fairly highly evolved but not the highest kind of plant that we would know. And that's true in every category here. The phenols that are present are pretty much the ones that you would predict if you just took a grab bag and said, all right, the most common phenols are these, and this is a somewhat woody plant of a dicotyledonous background, therefore we'd expect these. That's pretty much what you've got. So the grapevine is, in that sense, very normal and nice. It's kind of sad in a sense that it has nothing unusual in it that we can brag about, but maybe that's very good because it means we needn't worry about unusual toxic effects or unusual nutritional problems that relate to the components from grapes. Now, referring to your notes, in the benzoic acid group, salicylic, parahydroxybenzoic, vanillic, syringic, gentisic, gallic, protocatechuic, gamma-resorcylic, and metadigallic have all been reported, but some of these are marked as doubtful. For instance, salicylic acid is probably either an artifact, and certainly an artifact if it's over about 2 milligrams per liter, or an unusual degradative product. It seems unlikely that salicylic acid is going to be present in a truly natural sense in grapes, and this has been a big argument that goes back a long time. There's a bibliography in Portuguese about 1905 that lists something like 100 references to people studying whether or not salicylic acid occurs in wine. And the reason is that salicylic acid was added as a preservative for a long time. You know, and it's sodium acetyl salicylate is the aspirin. The idea was that, you know, it's perfectly innocuous. We take it as aspirin, so why not use it as a preservative in wine? So it's kind of like the DEPC story on an earlier date. It seemed reasonable to put it in, so they did, and then the argument came, but it's not a natural component, and then they said, oh yes it is too, and they argued for 50 years about it. Well, it finally boiled down to, if it's an artifact, if it's naturally present, it's certainly at a very tiny level, and if it's present at any reasonable amount, certainly somebody added it, or the barrel that the wine was in had wine in it that had such an additive or something along the line that made it be a contaminant. This is somewhat important from another viewpoint, because if we had the orthohydroxy type compounds, then it would be more reasonable to have coumarins. And since coumarin itself, for instance, has been banned as a food additive on a toxicity basis, liver damage basis, it would be kind of nice if we could eliminate the presence of the orthohydroxy derivatives, including coumarins, from wine. And we'll come back to that shortly. The presence of a number of these benzoic acid derivatives in the product that is, quote, released, unquote, by treatment with sodium hydroxide is quite bothersome, in that you cannot account for some of these compounds in the natural phenols that are isolated with less drastic treatment. Therefore, the benzoic acid derivatives have to be generated from something that's present. Well, it's been the habit to call these bound forms, but I don't think that's quite fair. And one compound that particularly incenses me, if you like, is gentisic acid, this compound, which has been proven to be present in alkaline-treated wine residue. Now, this alkaline treatment involves, say, reflux with 5% sodium hydroxide for 10 hours, or this sort of thing. Well, now that's that's liable to produce almost anything, it seems to me. And when you find gentisic acid in that kind of a product, and then say, as has been said, that of the total of these benzoic acid derivatives would run from 50 to 100 milligrams per liter in red wine and 1 to 5 in white. Now that in itself is significant, I think, because the whites contain very little flavonoid material, and it suggests then in the red wine you're degrading flavonoids to give this kind of product. And for instance, how in the world would you derive this sort of compound biosynthetically? You would expect the carboxy group to be para to the OH, so you'd expect it to be here, you see, or the OH to be here in any case. So, but that doesn't happen. So what I am proposing is that under the drastic conditions that are present, you have broken down a flavonoid with a carbon in this position to the benzoic derivative, and in the process you've gone through the NIH shift so that you've hydroxylated, inserting a hydroxyl group in the position where the carboxyl was and moving it to an adjacent ortho position. Now again, this is a hypothesis that hasn't been demonstrated. You can realize why. It would be easy to do, but very costly and time-consuming. You'd have to produce the proper cinnamate derivative or flavonoid derivative with a mark in the carboxyl and a mark in the ring, or a mark that would be useful in the position that the leaving group was going to leave. And your yield would be tiny. You'd start with a very high content of radioactive material and end up with a small fraction of that in the derivative, and it just hasn't been worth doing to anybody yet. But I believe that it's going to turn out that these compounds do not occur in wine naturally or in grapes, and this very drastic, quote, hydrolysis, unquote, is doing much more than simply releasing ester and glycoside bonds. So most of these compounds in the benzoic series would be artifacts. The only one that probably is not, or ones that are probably not, would be gallic acid and vanillic acid. Now, gallic acid would come easily from epicatechin gallate, and vanillic would come easily by oxidation from vanillin, which occurs as a barrel extract at least. So vanillin does occur in oak wood, and if wine is stored in wood, then vanillic acid and vanillin would be expected to be present, are known to be present. The cinnamic acid derivatives have to be present because the derivatives that are present in the flavonoid group would require the presence of the cinnamic acid derivatives. Both shikimic and quinic acids have been found in grapes, and there are at least 4 different cafe-oil quinates have been reported. Now, remembering that there's 4 OH groups that can be esterified with caffeic acid, and there is the possibility of dicafe-oil quinates, then certainly 4 seem possible. This is, however, disputed, and Pascal Ribeiro-Gaillon has one very good paper. I'm not questioning his results. He does have a paper where he showed that in grape skins he was able to isolate cafe-oil tartrate rather than cafe-oil quinate. Now, this is interesting from 2 viewpoints. Of course, the grape is a fruit with a relatively high amount of tartaric acid. Most other fruits don't have appreciable tartaric acid, so it might be reasonable if tartaric acid filled a role of this kind in the grape, and when it's not been widely found in other plants. It has been reported in a few others. Seems to me rosemary, anyway, some other plant, a cafe-oil tartrate analog of chlorogenic acid has been reported. On the other hand, and I asked Pascal this myself, why did he pick grape skins? Because we have shown that after his work, we showed that the cinnamic acid analogues are present mainly in juice and not in skins. So the chlorogenic content of the berry in total would have to, or the caffeic derivative, including chlorogenic, would have to be very low in the skins in proportion to the rest, whereas it would be very high in the juice. And also there is a considerable possibility of artifact production because and he did use red grapes again, which seems rather odd because if you're looking for a colorless compound, why contaminate it with a lot of other phenols from a red grape? He did use red grapes, and the acyl groups on the red pigments are mostly caffeic acid, and if you're going to have a high acyl anthocyanin pigment content in the presence of tartaric acid, and then do an isolation, it's quite possible you may produce some esterification of the caffeic to the OHs of the tartaric acid. So I'm not convinced that it's not an artifact. On the other hand, his implication is that the caffeic derivatives are not in fact chlorogenic acid, but rather tartaric acid derivatives. And at the moment, the question can't be resolved. I've had a couple of students that I've asked to work on this point, but they were all undergraduates and didn't stay. I mean, they were near finishing their work, and they didn't get far enough along to really prove the point. What I want to do is isolate a sufficient amount of the caffeic derivative from juice to definitely prove whether or not it is a tartrate or a quinate derivative. Fairly easy problem, but a little laborious, and it hasn't yet been done. Paracumaric ferulic derivatives occur as similar type to the chlorogenic caffeic derivatives, and these are found in wine, and interestingly enough, these can decompose to the benzoic derivatives, and that comes back then to the comment on benzoic acid. If you add cinnamic acid to wine, which ordinarily doesn't occur, you do find benzoic acid on a paper chromatogram, and we have never found orthocumaric acid present. No one has in grapes, therefore again the coumarins might appear doubtful. And the total of all cinnamic derivatives in Gallic acid equivalents would be around 200 milligrams per kilogram of berries. Now these of course are non-flavonoids, and other non-flavonoids have been reported, and some of these appear to be degradation products, like for instance metacresol has been reported in botrytized grapes, but not in normal grapes. So it would appear that that's a breakdown product from the growth of botrytis. And the vanillin, sinapahaldehyde, and so on from the oak barrel would be, again, sort of an artifact, but do occur in wine, but probably not in grapes. Although the reason they occur in barrels is because of the lignin generation and breakdown. In other words, they're either traces left over from formation of lignin or breakdown products of lignin. And since lignin is found in grape skins and grape seeds in a small amount, than they could be in wine, even if it hadn't been in the barrel. The lignin of grapes hasn't been studied very much, but it has been reported that the lignin of grape skin has no methoxy content, whereas that from seeds had 4% methoxy. Now that's somewhat significant because the inference in my mind is that how does one study lignin? Well, you study lignin by dissolving everything that you can away and leaving the lignin behind. Lignin is so inert that you dissolve out the cellulose, you dissolve out this, dissolve out that, and what's left you'll call lignin. Well, if you started with grape skins and did that sort of thing, a good deal of what you had left would be phlobophenes and polymeric tannins and not, in fact, lignin. And it's very difficult to tell the two apart. They're both insoluble, brownish polymers and phenolic nature and so on. So in my opinion, the so-called lignin that's been studied from grape skins was mostly large molecular weight tannin rather than lignin, whereas, and that's indicated by the lack of a methoxy content. If it were lignin, you certainly would expect it to contain considerable methoxy content, and that from seeds then must be true lignin, and certainly chewing up a grape seed would convince you that it ought to have lignin in it, whereas the skin you wouldn't be so sure. We've mentioned the ellagic acid story, and it has been reported both from grape leaves, juice, and wines, but notice I have question marks. It certainly is present in wooden barrel extract because ellagic tannins have been reported in oak wood. And although they haven't been studied, they probably occur also in the redwood and other wooden container materials. I've mentioned the fact that it could be an artifact from the conversion of gallate esters, and certainly no glycoside of ellagic has been found. So chebulagic acid, or one of these glycosidic derivatives that is usual in the ellagic tannins, have not been found in grapes. In fact, we have good evidence now that the tannins are essentially exclusively condensed tannins in grapes, whereas they are exclusively or nearly so hydrolyzable tannins in oak barrel wood. Therefore, you can use this as a measure of oak barrel contact. The coumarins in wine, I said we'd come back to, now is the time. Esculeton, umbelliferone, scopoleton have all been reported in wine, but the evidence is that they're probably artifacts for a number of reasons. The lack of orthohydroxy analogues that would produce them is one thing, and the other is that if you chromatograph such a thing as chlorogenic acid on paper chromatograms, you do generate these compounds in the course of the paper chromatography. So that what happens is by test tube reaction, you will convert the caffeic analog to the cis, and apparently hydroxylated in either this or this position, and then the lactone ring will form, making either this compound or this compound, and I tend to forget which is which, but I believe this one here is esculeton and this one is daphneton. In any case, both are found, so it appears to be a random chemical reaction that occurs on filter paper in the presence of air, and a trace of iron, which catalyzes the hydroxylation reaction and produces the artifact. So again, I feel good about this because I wouldn't want to be worrying about drinking a fair content of coumarins or eating them if I ate grapes or drank wine. And I think the fact that there is a tiny amount present, but it's an artifact, very likely, is somewhat reassuring. In this group also would be yeast derivatives. Tyrosol is the most obvious one, and tyrosol is the equivalent of a fusel oil that is produced from tyrosine. So if you decarboxylate and oxidatively deaminate tyrosine, you'll end up with tyrosol, which is the ethyl alcohol side chain on para-linked to phenol. And that tyrosol analog is found in wine in fairly high amounts, up to around 40 milligrams per liter. It's also found in anything else that's fermented. So it's been reported in beer, fruit wines, and so on. And so anything that has tyrosine and has yeast growth, or if it doesn't have tyrosine, the yeast might make it, is going to have tyrosol. And this has some importance because compounds of this type, particularly tyramine, have some physiological activity in humans. So tyramine, which is also high in cheese and does, I believe, occur in wine. I think Mr. O has found it along with histamine. Tyramine and alcohol tend to go together and produce hallucinogenic effect and what have you. So this may be a component of some significance in the physiological effect of fermented beverages. Since it doesn't distill easily, it wouldn't be in brandies to appreciable extent, but it would be in wine or other simple fermented solutions. And as analogues, it's been shown that paracumaric, ferulic acid, and probably caffeic and others produce derivatives like 4-ethylphenol and 4-ethylguaiacol and the analogues in that series, the vinyl analogues. So that from paracumaric acid, for example, we get this compound and the double-bonded vinyl analog from yeast action on paracumaric acid and similar compounds of the other members of the cinnamate series. And this is also found in compounds of this kind found in smoke, and so sometimes a sort of a smoky or pyrolyzed or heated flavor might possibly relate to this. That's carrying things a bit far. It certainly hasn't been proven that that's the origin of such flavors, but it has been proven that these compounds are produced by the yeast action on the natural phenols of grapes. By the same token, you should be able to find, but haven't been found, they've been found in other fermenting solutions, but not in wine, the lactic analogues and the other analogues in the series. So, in other words, reduction of the cinnamate group to give a lactic acid side chain and a keto group, a pyruvic type analog, should also occur, and no doubt if they're hunted for, they could be found. This may have some significance because the phenylacetic type compound, the compound with no OH, and the this compound, no, excuse me, phenylacetic acid, and ethyl phenylacetate are rose smelling compounds, and there are occasional wines that have a rosy odor, and it's been reported in champagnes that if you put in extra phenylalanine, then you do produce more of a rosy character in the champagne. Again, a lot of this work is Russian and other types that I don't necessarily believe in, and in some cases when we've tried to verify it, it hasn't turned out to be so. But it seems so logical, it may be that we just haven't gotten the conditions right, and at least it's the kind of thing to keep our eyes open for if we do hit such a especially rosy wine. I remember some white Rieslings that we had through our departmental taste panel quite a number of years ago that were very overripe, and they definitely had a sort of a wilted rose odor that was rather characteristic and strong, and we didn't know, still don't know today, why that group of Riesling was so different from usual, but maybe in an odd yeast or something of that sort. Whatever, it does happen on rare occasions apparently. This then brings us to the flavonoids, and the flavonoids would again be the usual series within the flavonol series, camphorol, quercetin, and myricetin. And I've mentioned that the myricetin has been reported to be absent by Pascal Ribeiro-Gaillon in white grapes, but present in red, whereas the other two are present in both. The catechins are reported, and the best identifications have been by Kathy Sai-Su, who worked with me some years ago, and this is a chromatogram from grape seeds, and the three main spots here are epicatechin, decatechin, and epicatechin gallate. And this has an interesting corollary that I'll come back to, so I'll leave it up there. We haven't yet identified all these other compounds, but we think this is epigallocatechin gallate, and there would be two more that are too faint to really see of the gallocatechin and epigallocatechin. So there'd be another trio like this one in this region, and we do see it on some chromatograms. And then this group includes leucoanthocyanins, but it also includes compounds that don't turn pink under acid treatment. And these would be largely the tannins, which are also leucoanthocyanin types. And we do want to complete identifications not only on the grape seeds, which are a little less important maybe from a wine viewpoint, but more and more examples from grape skins. But this is a fairly time-consuming work, and we haven't gotten as far as we'd like to with this yet. Now, the point I wanted to make with regard to epicadic and gallate, now this is a chromatogram from very underripe grape seeds. So if you pick grapes green before the sugar content is maybe over 10, very underripe from a winemaking viewpoint, you'll find epicadic and gallate is very high, just as it is here. It's about equal in amount to the other two members of the trio. If you then let the grapes get ripe, this will almost disappear, and by the time you make wine out of it, if you get grape seeds from wine pumice, you'll find this is absent and you can't find it at all. Well, you might say epicadic and gallate is going to hydrolyze then, and maybe it's just converting to gallic acid and epicadicin. Well, if that's true, the epicadicin spot ought to grow as this diminishes, and the gallic acid ought to appear. Now, unfortunately, gallic acid in this chromatographic system runs exactly where catechin does, so we had to do other studies, but we were able to show that there is no, as the epicadicin gallate disappears, gallic acid does not appear. And so there is no free gallic acid in the system at any time, at least not a reasonable amount. We couldn't detect any. That being so, then this again comes back to maybe epicadicin gallate and compounds of that type are serving as a source of gallate for later use. And it's intriguing that the formation of delphinidin and malvidin begins to occur about the time that this begins to disappear. So that the formation of the trihydroxy B-ring compounds at verazum and later is about related to the disappearance of this compound. So again, it's an interesting mystery that we haven't yet clarified, but we think we're on the track of something biochemically rather interesting if we can finish working it out. The leucoanthocyanins, or anthocyaninogens, are nearly exclusive cyanidin generators. The leucodelphinidins have been reported, but if they're present, they're much minor amount compared to the cyanidin generators. So by and large, in grapes and wine, the only one we need worry about are those that generate cyanidin, and it's interesting that no pelargonidin generators have been found, and since pelargonidin doesn't occur in the pigments themselves, that would be reasonable. I'm not implying that the anthocyanins are generated by biosynthesis from the leuco compounds. Quite the contrary. It's been proven that that's not so, but at least you'd expect the two to occur together if that were so. The flavan-3,4-diols as monomers apparently don't occur in grapes, so we have not been able to find the, strictly speaking, leucocyanidin of the single monomeric structure. But the dimers have been found, and larger units have been found. So far, it appears your notes say 2 to 8 unit polymers, but as I mentioned the other day, it appears that it's mainly 2, then possibly 3, and then 6 or 8. So in other words, there does appear to be a gap and suggesting enzymatic control and not just random synthesis. These dimer and trimer forms do include catechin as the terminating unit, so that dimer would ordinarily be one leuco-type unit and one catechin-type unit. And depending on how you convert it to the anthocyanin, you'll get more or less yield of the leucoanthocyanin. I did want to bring up a couple of other points while we have the slide on, so I'll go to... if we take all the phenols, now let's talk briefly about the quantitative aspect. If we take all the phenols from a grape, it would total something like these figures. And notice that the red grape totals about 5,600, and this is an average of several varieties, and the white grape's about 4,000. Well, that difference is due to two major differences. One is that apparently the ability to make red pigments is lost in the white grape, and I was able to say up until this year there are no wild grapes known that have white berries, that the white berry condition is a lost mutation controlled by man, or at least propagated by man. I've read just recently of some South American wild grapes that do have white, pink, and red berries in the wild. Now, I'm not so sure the white aren't underripe. If that's so, then that wouldn't defeat my story. But at least by and large, all the grapes that we know that have been used to make now commercial valuable grapes, the wild condition is red, and the ability to make anthocyanins is lost in the white. And therefore, it's interesting that the difference between the total phenol in the skin of the red as compared to the skin of the white is about the same as the difference in the total phenol overall. So in other words, the grape, having lost the ability to make the red pigment, doesn't make anything else. It just loses that ability, and the total becomes less. Now, another reason that the red grape tends to have higher phenols, it tend to be smaller and seedier berries, and since the seed is added in, notice it's higher as well. Thompson seedless can be included with the whites, for example. This is another reason that generally the reds look to be a little bit higher. Well, we'll have to quit at this point. If you haven't turned in your